Perfect. Yeah, it's great to have this opportunity to show you how you can use NMR to dissect the structure of large protein assemblies. So I'm from the Utrecht NMR group in the Netherlands, and there we have expertise in solid state and solution state NMR, computational structural biology and molecular biology. So really a great place to do integrative structural biology. And here you see also a picture of one of our NMR instruments. And actually this one will be moved later this month to make place for the most powerful NMR magnet available these days, that's operating at 1.2 gigahertz. So what I find fascinating and why I do this integrative structural biology is that life at the smallest level all comes down to these protein molecules that have to find each other and have to assemble into some kind of complex in order to perform a task that they can only do if they assemble correctly. Like in this case here, where you have 4 components that come together in order to ubiquitinate only one particular lysine residue in the nucleosome. So you may think, yeah, okay, what has NMR to do in this area of very large systems? Isn't that only for small protein domains? Well, that's certainly not true. Here you see 4 examples from literature where people have used mainly structural information coming from NMR to determine the architecture and the structure of complexes well above 100 kilodalton up to half a megadalton size. So the general strategy to do this is that you have, of course, your subunits of the complex, and you need to know the structures of these subunits. If you do not know it, then you will have to determine those structures first. The subunits can be anywhere 10 to 100 kilodalton, let's say. And what you then use the NMR for is to get information, structural information on the interaction. So that can be very simply defining which residues form the binding interface, or it can be more quantitative to measure really intermolecular distances across the interface or the relative orientation between the subunits. And once you have this information, then you combine that and perform some kind of integrative modeling procedure to come up with the structure of a complex. So the great thing of using NMR here is that, of course, this is a non-destructive, non-invasive technique. You don't need to crystallize your complex. And NMR is a quite versatile technique. You can apply it if your sample is in solution, in a sediment, in some kind of gel, in the solid, or even in a cell. It's also actually a very robust way of characterizing interactions, and it's sensitive to essentially all interactions, from the very strong to the very weak. Well, having said that, there's of course also some challenges when you perform NMR studies on such systems. So first and foremost is practical considerations. You need to isotope label your protein. And you need to have milligram amounts of your protein. And finally, it's also important to realize that NMR data will never give you direct access to atomic coordinates, but you will always have to rely on some kind of modeling procedure to translate the information into a structure. So why is NMR actually such a good technique to study protein interaction? Well, and the NMR signal that we observe comes from an intrinsic magnetic moment in the nucleus of an atom. And that is just extremely sensitive to the electron cloud around that nucleus. And that gives us the chemical shift. And that gives the fact that NMR has really an incredible chemical resolution. That means that we can see distinctly the signal of one particular methyl group, for instance, this valine here, in one particular histone in the nucleosome. And if you then add a protein, and if that's close to that particular atom nucleus, then you will see that this NMR signal will be perturbed. It will, in principle, get a change in the frequency, a chemical shift change, which we call a chemical shift perturbation. But also, especially if you form a very large complex, you will see an effect on the intensity, that in the bound state, the peak is actually broader and has a lower intensity. So practically what you will have to do if you pursue an NMR interaction study is, first, of course, you have to make your sample, which means that you have to isotope label your protein with the NMR active versions of nitrogen and carbon that are 15N and 13C. And also often you have to incorporate deuterium as well. Then in the end you get an NMR sample which is anywhere between 150 and microliters and 500 microliters with, let's say, 100 micromolar of your protein. Now in the end it's all about the quality of the spectrum, and that determines whether you will be successful to study that. And it will depend, of course, on the buffer conditions, the pH, temperature, etc., but also on the type of labeling strategy that you employ. You can use uniform labeling, where you replace all the nitrogens with 15N and all the carbons with 13C, but you can also do that only in parts of the protein or only in specific residues or in specific chemical groups. And that can make really a huge difference, as you can see in that figure there, where you can change from basically one big blob into really a beautiful, well-resolved spectrum. Now, in order to use such a spectrum, you need to know for every signal to what atom in the molecule it corresponds. So that's called the assignments. Sometimes you can just simply, this has already been done, and you can simply download it from the web. But in other cases, you will have to perform dedicated experiments to obtain these assignments. And once these things are in place, then you can perform the interaction studies. Now, there's different ways to do that that will give different types of information, like I said, so the interface or distances and orientations. And what I will show and highlight in the rest of the talk is just the titration experiment, which is the most simple, easy, robust, and fastest experiment that you can do, in which you simply add an unlabeled interaction partner to the labeled protein. And this will give you the interface residues and will also give you an idea of the KD and the off rate, or the lifetime of the complex. If you do that for high molecular weight systems, there are a couple of things to take care of, because you run into a fundamental problem. So if you have a very large protein or assembly like the nucleosome here, you have a lot of hydrogen atoms, and that just gives a situation that the spectrum is very crowded and that each individual signal will be extremely or very broad. So there's two solutions, two main solutions to this problem. First, in solution NMR, what we do is just remove all the protons by deuterating the protein, except for the methyl groups. And then we do a special type of NMR experiment that we call the methyl-Trosi experiment, and that gives you, again, a very nice spectrum with very sharp lines. And the other approach is to go to a solid-state NMR, and in that case you also reduce your protons often by deuteration, but not so drastically. And what you then have to do is to spin your sample extremely fast, and then that will also give you sharp lines again, so that you can do high-resolution NMR studies. So for the system of my own interest, that's the nucleosome, these two approaches work very well, because you can make these histone proteins individually, you can apply the appropriate labeling procedure, and then reconstitute these nucleosomes and put them in a solution or in a sediment to do a solid-state NMR in this latter case. And both approaches give really excellent high-quality spectra, for which we already obtained all the assignments. I want to illustrate these NMR approach to study macromolecular assemblies with two examples from my group. The first is on the ubiquitination of nucleosomes and the DNA damage response. So where these four components, an E3 ubiquitin conjugating enzyme, an E2 ubiquitin ligase, and ubiquitin and the substrate have to come together. And these three proteins together cause the specific ubiquitination of lysine 15 in H2A in the DNA damage response. And this project originally comes from Titia Sixma lab at the Netherlands Cancer Institute. And they tried to crystallize this interaction, but that really didn't work out because this E3 protein is frankly quite a shitty protein. So that's why we turned to NMR. In this case, the interaction between the E3 and the E2 and between the E2 and the ubiquitin is already pretty well known from other systems. So what it boils down to is to figuring out how the E3 interacts with the nucleosome. So that's where we used our NMR approach for. In this case, we used the solution NMR approach with the methyl group labeling. So what you see here is a spectrum, a fingerprint spectrum of a nucleosome in which H2A and H2B, two of the core histones, were methyl group labeled, and each dot here represents a methyl group. And we then just simply added this RNF E3 protein to the sample and record the spectrum again, and do it again and again and again to monitor these changes in chemical shift and in peak intensity. And indeed, we find that there are specific changes in the spectrum for methyl groups, and these all localize actually together on the nucleosome surface, indicating that this is the binding surface for the RNF E3 protein. This by itself is not enough to determine the structure. You need to have at least one more source of information, and that's the binding surface on the RNF, on the E3 protein. That we obtained from mutagenesis. So we knew that two arginines were crucial on the RNF side. And that still can be enough if the structure is just right, that there's only one way to fit them together. But in general, you will have multiple options to put these two surfaces together. So you also need to have some kind of orientational information. And this we got from cross-linking mass spectrometry, where we could identify one intermolecular cross-link between the E3 and histone protein. And with all these three sources of information together, we could determine this integrative structure of the E3 nucleosome complex. And that shows really nicely how this arginine-rich helix of the E3 fits snugly on the nucleosome surface, contacting the so-called acidic patch. So this structure is deposited in the PDB-DEF database, for which you will also have a talk later, I believe, in this seminar. So this is an example. Actually, this is step one, the E3 nucleosome complex, and what we then did is to extend this to the quaternary complex using the structural information based on homology on how the E3, the E2, and the ubiquitin sit together. And this structure shows very clearly two things. One, the origin of the specificity. The E3 binds such on the nucleosome that the E2 and the ubiquitin are positioned directly above the active site, above the target lysine. And second, it explained the observation that the presence of the nucleosome actually accelerates the ubiquitination reaction. And that is because this large steric presence of the nucleosome prevents opening of the E3, E2 ubiquitin complex, which is not catalytically active. Okay, this was an example where we essentially started from crystallography and moved to NMR. The last example that I want to show is actually the opposite way, starting from NMR going to crystallography. And this is a study of a histone chaperone called APLF, trying to figure out how it interacts with the core histones. We did, again, we started from an NMR titration experiment, adding the core histone H2A, H2B complex to APLF, looking at the NMR spectra, seeing peak intensity changing, mapping the binding site. And to our surprise, we found that this protein can actually also bind the other core histone complex, which is called H3H4. And these two histones complexes are bound by adjacent and non-overlapping binding sites. Then if we do the reverse experiment and look at where do they bind the core histones, we see that it actually binds really to a well-defined spot on the histone. So this is really looks like a valid interaction, and this suggests that this chaperone can actually bind all core histones simultaneously. So from a series of experiments, we could indeed validate that. And from NMR, we see that if you add all the 4 histones together, we lose the binding or the signals in both binding domains. From ITC, we see that this is really a very tight interaction when you add all the histones together. From native mass spectrometry, we could determine that the mass is such that it corresponds to a histone octamer with 2 of these APLF molecules bound. And this was fully consistent with solution SAXS data that also suggested the size is compatible with the structure of a native histone octamer. So using all this information, we were in the process of modeling this structure. But of course, here the problem is, what is the structure of one of our subunits, the APLF? This is an intrinsically disordered protein for which we certainly do not know what structure it will adopt in the bound state. So here we instead use this integrative model to come up with a good design of constructs to try for crystallography that would exclude all the floppy bits and really focus on the interaction. And this worked perfectly well, resulting in this 2.3 angstrom resolution structure of a fragment of this APLF chaperone domain, showing how it teeters together these 2 core histone complexes into 120 kilodalton complex. So yeah, with that, I would like to hope that I've shown you clearly how you can use NMR to study macromolecular assemblies, different approaches available, and I hope also that you will consider to use it in your own project. And you don't need to be an NMR expert or to have the NMR machines. You can apply for access through the iNext Discovery program. Okay, thank you.